Greetings from Uganda, East Africa, and greetings to the team. Thank you for the opportunity. Gratitude to the Center for Public Health and Zunoz's team for giving me such a platform. My name is Christopher Kadala. I'm a Ugandan, a researcher. I'm currently pursuing a master's degree in One Health and Infectious Diseases. Additionally, I hold a bachelor's degree of science in public health and also a diploma in livestock health science. Today, I'll be presenting on the Zirimerin LARVEX system, an integrated One Health approach targeting aquatic larval stages of disease vectors, including trematode larvae. Vector-borne diseases continue to pose a major global health challenge, particularly in low-resource settings. While much attention is given to adult vectors, the aquatic larval stages, especially mosquitoes and trematode larvae, remain insufficiently targeted. This creates a persistent cycle of transmission across human, animals, and the environment. And of course, since we have earlier talked about the One Health approach, our research is mainly anchored in the One Health mechanism of disease control and prevention. Current control methods face several limitations, including increasing resistance, environmental toxicity, low residual activity, and lack of integration across sectors. Additionally, most interventions are species-specific and fail to address multiple pathogens simultaneously. When I talk about that, I mean that the current interventions cannot target different species, and mostly in Uganda, they target only mosquito larvae. And according to documentations, there are no interventions that can target both mosquito larvae and trematode larvae, plus other disease-causing vectors in the transmission cycle of aquatic stages. The objective of this work is to develop an eco-friendly multi-target larval control system that effectively disrupts aquatic stages of vectors and parasites within a One Health framework. The Zirimerin LARVEX system is designed as an integrated larvicidal approach targeting aquatic environments. It utilizes plant-derived bioactive compounds to disrupt larval development and survival. Importantly, the system is designed to act on both insect larvae and trematode stages, addressing a critical gap in current control strategies. The system works through bioactive compounds that interfere with key biological processes in larvae, including membrane integrity, metabolic pathways, and developmental progression. This leads to larval mortality or failure to mature into infectious or adult stages. The mechanism is selective and environmentally considerate. So, since it is derived from our botany, it renders low or no environmental risks, and it is conscious to non-target organisms since it is applied in water bodies that may include fish and other aquatic organisms, and since it can also be applied in the water bodies shared by humans and livestock, it renders no effect or negative effect to the livestock systems or human health. The development process involves the extraction of plant-derived compounds, followed by laboratory-based larvicidal assays. These are used to evaluate efficacy against target organisms and against target organisms under controlled conditions. The system is further designed with scalability in mind for field deployment in endemic settings. So, this system, as I told you earlier, one, it has no or low environmental effect. It is environmentally conscious since it is derived from our local plants with bioactive ingredients. Secondly, it has no targets to the non-target. Has no effect to the non-target organisms due to its selectivity. And lastly, it employs the photodynamic or photosensitive mechanism, whereby it needs late activation to offer sustainable efficacy. Preliminary findings and conceptual projections suggest strong larvicidal potential with the ability to reduce water populations and interrupt transmission cycles. Additionally, the system supports environmentally sustainable control strategies. The Zirimerin LS aligns strongly with the One Health approach by simultaneously addressing human, animal, and environmental health. By targeting shared aquatic environments, the system reduces disease risks across multiple hosts while minimizing ecological disruption. This system represents a shift from single target interventions to integrated larval control. Its innovation lies in combining ecological safety, multi-species targeting, and applicability in resource-limited settings. According to Uganda and Africa at large, we have a strong burden and a persistent burden of vector-borne infections, and these are normally prevalent and endemic in the low and mid-income countries. For example, our Uganda, and in those communities with the low accessibility to resources. In conclusion, the Zirimerin LARVEX system offers a promising sustainable solution for aquatic vector control. It has the potential to strengthen disease prevention, offers strong effort through an integrated One Health approach, and since the World Health Organization, the Food and Agriculture Organization (FAO), and the World Organization for Animal Health launched, together with the UN agencies, launched the One Health approach to disease control. I, Christopher, also, in partnership with and collaboration with my team and my colleagues, we want to also pioneer and champion that One Health approach in Uganda. In addition, lastly, I and other scientists are looking at zoonotic health as a priority in our research, because you can see that Rift Valley fever also can transfer or can be spread from animals to humans. I would like to acknowledge, as I conclude, the organizers of the Center for Public Health and Zoonoses, together with the selection committee, for giving me the opportunity to present a recording of my research and my study, my work to you people. Thank you so much, and I'm grateful for this wonderful opportunity. Thank you for your attention. I remain Christopher Kadala from Uganda, a researcher in One Health, targeting most of the zoonotic diseases, and in this study I mostly targeted Rift Valley fever, malaria, dengue, and trematode spread infections, and the parasites like liver flukes and the schistosomiasis parasites. Thanks so much again. Good day and goodbye. I look forward to hearing positively from you. I remain Christopher from Uganda, Africa.